Okay, thanks for attending our special virtual advanced mass spectrometry seminar today, and hope you have prepared beer and snacks by yourself so you can enjoy it in our seminar as the on-site meeting. So today we're going to talk about XMS Viewer, and we want to see how it can facilitate our raw chromatogram examination and biological interpretation together. So I will show you the design concept of XMS Viewer and the application of the software using the real-life data. So in the future, maybe you will have a collaboration with BayBioMS on untargeted metabolomic studies, then you can know what you can expect from us. Yeah. Okay, we go. So untargeted metabolomics, I think, is getting popular right now. If you search the term untargeted metabolomics, the number of related publications increases really dramatically in the last few years. Starting from 2004, there were only 2 publications, but last year there were more than 700 publications. Actually, more interestingly, if you search the term metabolomics, the total number of publications is much bigger, but the trend, if you look at the trend, the increasing is not as deep as at the untargeted metabolomics. So untargeted is really a hot area in the field. So recently, we also have a few untargeted metabolomics projects. And also during our data analysis, we realized that the softwares currently available are not so easy to work with, at least to our purpose. So we decided to implement something by ourselves to fit our purpose perfectly. So XMS Viewer. So XCMS Viewer is a collection of R packages, and it can be used to process, analyze, and also visualizing the result. So it is based on the XCMS package. It is a very well implemented package by Smith and coworkers many years ago. But with XMS Viewer, we greatly extended its functionality and particularly focus on the interactive exploration. So it has some unique features in my point of view. And the first is the intensity matrix centric. So we say intensity matrix. Intensity matrix is a matrix where the rows are the metabolites and columns are the samples. All the values in the matrix are intensities. The intensity indicate how abundant a metabolite is in a sample. So this metric is very important because all the downstream analysis will rely on this matrix, for example, the t-test, PCA. So it is the input for the downstream analysis. This concept is very well accepted in the transcriptome or proteomics study, I think, but not so well in the metabolomics. I think that could be caused immaturity of the data processing in metabolomics. Therefore, many of the softwares focus on the quality control or some single sample exploration function, but not consider all the experiment as a whole. Yeah, that's the intensity matrix centric. Also, in the XMS Viewer, we have a comprehensive annotation of the data using both MS1 and MS2 information. Also, the database we use could be customized according to the need of the project. And also, the interactive exploration is also a unique feature of XMS Viewer. So because most of the time we want to use untargeted metabolomics to generate hypotheses. We need to do this, we need to explore many metabolites. For every metabolite, then we need to check their chromatogram. Are they looks good? MS2 peaks, are they map well with known metabolites? or if the metabolite is top ranked or selected by the t-test, we need to check the fold change, we need to check the p-value, many different things. A decision can only be made when we evaluating all these informations. So doing this manually, I think is a pain. But interactive visualization gives you a way, just a one click, then you can see all the information. So it's really facilitate your hypothesis generation, particularly when you need to explore a lot of metabolites. And the next is flexible statistical analysis, because this is seamlessly integrated in the R environment. All the flexibility in R are inherited by XMS Viewer. And also, because we have a lot of collaborations, we need to think about how we can deliver our result to our collaborators. So XMS Viewer can be delivered as a standalone software. So you can double click and then open a page in the browser, then you can explore your data. Or it can be hosted as a web application. So if you think about publication, you can give a link, then all the reviewer and readers can explore the data in the same way as you do. The last feature we consider is the scalability, because now there are more and more experiments are focusing on some complex phenotypes, for example, defined subtypes. In this experiment, we don't have like typical experimental versus control design, but what we have is a large number of sample and complex phenotype. So we can achieve this by modifying the XCMS pipeline, by processing files individually first and then combining them together. So by doing this, we can increase the number of sample size we can process, but don't increase a lot on the memory we require. So next, we move to the data processing pipeline. So if you work with XCMS, you're already familiar with some of the steps of XCMS Viewer. So if you look at the top 3 row on the left panel, so data acquisition and format conversion, followed by the peak detection and feature grouping. All these steps are done by the XCMS package. At the end, what we have is an intensity matrix we talked a few minutes ago. And when we have that, we will use the XCMS pipeline to annotate the features we identified based on the MS1, the mass information, and MS2, the fragmentation pattern information. And then we can do the statistical analysis on the intensity matrix. For example, if you have two phenotypes want to compare, we can use t-test. If we have more than 2, we can use analysis of variance. Or we can also use some unsupervised method like PCA or PCA, right? The important thing is we need to find out some statistics, and that can be visualized in the 2D scatter plot. So you will see why this is useful later by an example. So XCMS Viewer will store all the results in every step, and then put them together so you can explore all these information almost at the same time. So next, let's look at one example. So in this example, we have 2 breeds of potatoes. We call them A and B. And so there are 6 samples in total, 3A, 3B. And what we know is B is more resistant to fungi or bacterial infection. And then what we want to find out is what metabolite makes the difference. Of course, using untargeted metabolomics. So this example is from our collaborator, but the biology I show here is not important because the data is unpublished. I don't show the most interesting biological results here. So we will do two things. One thing is targeted exploration. The other one is untargeted exploration. By doing targeted exploration, we can check the spec-ins for QCs. Let's assume that in this experiment, we have two spec-in metabolites, tyrosine and lysine. Yeah. And then we see how we can check them. Also using, sometimes you have a predefined list of interesting metabolites, and then you can use you can explore them in a targeted way. So untargeted way, mainly by the figures, and then you can see how we can work with interactive figures in the XMS Viewer. So now let's move on to the software. Okay. So this is the interface of XMS Viewer. And the first step to work with it is to look at the top right corner. You need to give the data. So in most of the cases, we have at least 2 data sets, and you can select them using this box on the top right corner. Here we select this XMS stats example pos. It may take a few minutes if you have many samples, but here it's rather quick. We have only 6 samples. So the first thing you see in the middle is what we call the feature table. So in the feature table, every row is a feature. We can assume every row is a metabolite, or is a group of metabolites. And the first column here is the ID of the metabolite, of ID of the feature. And second row is the possible annotations based on MS1 or based on the mass information. And here, please note that for every feature, we may have tens of annotations or tens of mapped metabolites, but here we only show 3 or 4 of them because of this space is limited. And the third column here is the QC column. Actually, all the features I show here pass the QC, so all labeled with a plus symbol. This column is followed by the MZMAT. MZMAT is the median of M over Z ratio. So it's the mass information of the feature. And the last column is the retention time, RT, retention time, the median retention time of all the features, of the features. Okay, so this is the feature table. And then you can see is the top left corner. Here I got phenotype or metadata. So this information is about the samples you measured. So here we have 6 samples. I named them files 1 to file 6. And the first 3 files are from group A. And the sample 4 to 6 are from group B. So we want to compare A and B. Yeah. So to explore the data in a targeted way, the easiest way is to search your metabolites in the search box on top. So you can search according to the range, search according to the M over Z ratio. But in this case, we only need to search metabolite name. Let's look at the first spec in. Let's say it's tyrosine. And then you click filter. So you see the table are filtered, and every feature here have been annotated by Terracing, at least the keywords Terracing. Actually, the first one you see Terracing here, and you can click on it. And there are some other information displayed in the window. But let's first look at the top right corner. The top right corner, I call it annotation. So this panel gives you the annotation information. There are 2 tables. Let's look at the table on the top first. This is the annotation using exact mass matching. So that means the MS1 information. Which metabolite can be matched to the mass we measured in this feature. So the top one is Terracing. Here you can see the name column, and also an interesting column could be score. The score indicates how likely the notation is correct. This score integrates the information of the cosine of MS2 spectra, the cosine distance of MS2 spectra, and also the adduct information. Some of the adducts, here you can see this is the proton adduct. Potent form of the metabolite is very likely to happen in the positive mode. But if you have something else, maybe have a formic acid adduct, then maybe it's less likely to happen. Then you give less score on it. The third information the score considers the delta information. This delta means the mass difference between the theoretical mass and the mass we measured. At least you can assume that the top ones are more likely to be the correct annotations. Yeah. Then we can look at the table on the bottom. This is called MS2 related to these features. So what we have here is we have MS2 fragmentation pattern of this feature. And also we have a lot of fragmentation patterns recorded in the database. We compare the fragmentation we acquired with the database. As an example, the comparison is done in this way, showing on the bottom, on the right side. This is we call the mirror plot. In this mirror plot, on the top, we show the fragmentation pattern in our experiment. On the bottom are the fragmentation patterns in the database. So whenever the mass of peaks are the same or smaller than 10 ppm, in this case, there is a green line. That means those two peaks are considered to be the same. And here we can see there are 6 peaks matched together, then it's rather high confidence, I guess. Yeah. Sometimes we only see a peak can be matched, and the mass of the peak is the precursor mass. Then actually it's not really, we don't really use the fragmentation information. Yeah. Okay, so this is annotation column. Then let's look at the left side, the bottom left side. Here we see 2 more figures. So the figure on the top is a bar plot. So every bar is a file, and the height of the bar indicates the intensity of the feature in each file. So this is a spike in metabolite. We want to check if all the samples, if we identified this metabolite Terracing in all the samples. Actually, no, because you can see file 2. The intensity of file 2 here is 0. And if we want to know why this is the case, we can click on this bar. It's very low. And then the corresponding file will be highlighted on the top table, and the chromatogram will be highlighted by the right color on the bottom. So you can see, actually there is a nice peak, but the peak is much lower compared with others. So it actually, in this case, the intensity is 0 because intensity of the height of the peak is too low. We use the threshold 1000. At least the 5 data points has to be higher than 1000 in an experiment to identify a peak. But maybe this peak, unfortunately, doesn't go across this threshold. So the intensity here is 0. And then we can click on the bars. Maybe we can now click on the highest bar to see which chromatogram it corresponds to. Actually, we can see the highest bar corresponds to the chromatogram the highest chromatogram. So the intensity here I think is okay. The next is file 6, the next highest chromatogram, and the file 4, the next highest, and then by file 1, and then file 3. So by doing this, you can evaluate how likely your intensity call is right or wrong. I think in this case, it's rather good. Yeah. Sometimes when you have a problem, you may don't identify the QC or the spike in for QC. What you have to do is you can use free chromatogram. So by doing this, you don't specify the name of the metabolite. What you need to specify is the retention time, or M over Z. M over Z here is more important. Let's assume that next spike in we want to explore is the lysine. Lysine has a mass of 146.19. And because this is a positive mode experiment, we can identify the proton version of the metabolite, that is 147. Let's give a range here, 47.3. So this range, M over that range, covers the proton version of lysine. We can apply the filter, and then it extracts the chromatogram. And of course, if you want to use the metabolite as a QC, it is good you have a rough idea what its retention time is, because in this case we have 2 peaks. You don't know which one could be the lysine if you don't know the retention time. Actually, this will be the lysine. You can zoom in here, and the peak is not very smooth, but I think it's also a nice peak. So you can check which sample have detected, which sample have not detected this QC, this spike in metabolite. Okay. Using the same mechanism, you can also explore the data in an untargeted way. If you have a list of interesting metabolites, you can easily explore 10 or few tens of them by searching different metabolite names. Next example, let's see how we can explore our data in an untargeted way. And now I remove the filter here. The best way to work with untargeted data is to work with the figure, because in this case, in this specific case, we are comparing group A and group B. What I did is I used the t-test to compare every feature in this table to see if A is higher than B or B is higher than A. We call this differential analysis. The differential analysis generates 2 important information. One is the p-value, tells you whether the difference is significant statistically. The second information is the fold change. So it's like effect size and how big the difference is between A and B. So by combining these 2 information, we often use a figure we call volcano plot. The volcano plot can be shown by here using the feature tag. This is a 2D plot, but here the x-axis is the retention time, the y-axis is the M over Z, and every point here is a feature in the table. And now we want to change this plot. The x-axis is a fold change. Here I called it MD, and the y-axis is the log transform, the FDR, false discovery rate. Of course, when we have a collaboration, I will tell you what each column means. Yeah. And if you used it, you will know by heart. So of course, the most interesting part in this plot will be the top left and top right corner. They have low p-value and high fold change. But because this data is unpublished, I won't click through those ones on the top left and top right corner. I think many of them are very interesting. Maybe our collaborators is doing some follow-up experiment on them. As a demonstrative purpose, I will select some points here. So it has less biological interest, but the mechanism of looking them are the same. Yeah, once you clicked on a point, so the same information, the annotation and the intensity information will be shown on the left and the right side. You can explore them in the same way. Something could be convenient is you change the intensity, change the way of showing intensity. So here, every file's intensity are shown by a bar. So because we want to compare A and B, it's better we use the group information. We can show something like a box plot. Yeah. So we can clearly see in this case, A is higher than B. Yeah. And we can check their chromatogram. Yeah, this is the same in the chromatogram, and we can see the notation here. It could be the neuromedin, but there's no MS2 information. So, of course, in most of the cases, you need to do this click the points many times. You will click through all of these points and to see are they make sense, and combining all these MS2, MS1, and the p-value, fold change information, then you can make an assumption, maybe find some interesting target for your downstream analysis. Okay, that's the demonstration of the XCMS Viewer. So we're back to the slides. So the last thing I want to talk is about the future extensions, very briefly. So I also want to integrate the zeros. Zeros will annotate more metabolites based on the MS2 fragmentation, I think. GNPS is a very nice online tool, give you the network based on the fragments and the fragments of metabolite. And now we are scoring the MS2 spectra purely based on the cosine distance. Maybe there are some other way we can score the MS2 information. Something else important is the pathway information. So for me, who lacks the background biological knowledge, it's always hard to derive a biological hypothesis by looking at the names of the metabolites. But if we have a pathway information, I think that would be much easier. So the last thing I thought about is to optimize parameter automatically, so we don't need to manually check all the parameters. Are they work good or work not so good? Yeah, so this can be automated, would be good. So all this can be split into small parts that are available for students to work on as a master student, bachelor student, or internship. So if you are interested in developing XCMS Viewer, so you can shoot me an email. We can discuss details in our meeting. So if you want to follow the development of XCMS Viewer, you can go to the GitHub page. I put it here in the slide. Thanks for attending our virtual seminar, and I'm happy to take your questions if you have. Karin?
This is a great presentation about XCMS Viewer. So people who have worked with untargeted data before and with vendor software, having all the information on one page is pretty awesome, because the vendor software you have to extract the peak first, and you have to search for the MS2, and then you compare it to databases. So this is a lot of work, and Chen's tool is pretty awesome that you have it all on one view and that you can dig through your data. So are there any questions from the audience? So there was nothing in the chat. I also don't know. I don't see if somebody raises his hand.
So you can unmute yourself, I guess that's also possible.
Yeah.
Or you can shout in the chat window, we can unmute you.
So maybe I start off as usual with the first question. So you showed your QC. So maybe comment what is behind the QC. So this is not our QC sample we measure. It's the QC you created, or?
Yeah, the QC. So every peak. So in the peak detection step, when we define a peak, it has to be like a bell shape, and the intensity needs to be high that it can be distinguished from the background noise. So QC actually check these informations. Here, I used a very simple QC. The intensity has to be higher than 1000, and at least 5 points has to be higher than 1000. Of course, another thing could be we fit a Gaussian curve to see how different our points compared with the Gaussian curve we fit. So this QC is done for every single peak. It's not a QC sample or QC spike ins. It's for QC of the peaks.
Thanks. So we actually got some questions now. So Manfred Repke is asking, or maybe you can also unmute him if he wants, if you can use the XMS Viewer for GCMS data. So we have not done it, and also so I'm running the GCMS in our lab. So we just have a low resolution mass spec. So I don't think that would work proper. But maybe Chen, you want to comment on GCMS and XMS Viewer?
No, unfortunately, I didn't check GCMS data on this. I guess very likely there will be a problem. But could XCMS process GCMS data? Do you know that?
So I tried that with our machine, but I could not convert the data into in the format I needed to have. And also the difference is, so in GC we don't measure MS2 spectra, we measure EI spectra. So in general, you already have the fragments in your chromatogram. But the advantage of GCMS data is that the spectra are much better comparable between different suppliers. So there are databases like NIST where you have a ton of EI spectra and you can compare them, and it's wherever you go, which machine you measure, it's always the same spectra. So this is quite nice. But I would also say it would require some work to make XMS Viewer compatible with GCMS data. So yeah.
But I think in the future, if we need, we can check some GCMS data. And of course, if you are interested to develop by yourself, you are welcome. Also, you're welcome to contact me. We can see how we can do this.
So does this answer your question, Manfred? If so, we move on to the next question from Sebastian Diekmann. He's asking if your tool would also work for multi-imaging data.
I think the same here. I didn't work with multi-imaging data. But I assume there will be a problem.
Well, I think our multi is again just low resolution, so we need mass accuracy to do the proper annotation of the metabolites. And the multi-imaging data also does not contain any MS2 spectra. So Sebastian, I think we need to use your multi-imaging samples, extract them, and run them on the UPLC QTOF, and then we can use the XMS Viewer to analyze your data. So I would say yes, if we do the workaround, putting your sample into the LCMS system, and then we can correlate your multi-imaging data with XMS Viewer, but we need to do the extra measurements. Okay, so are there, okay, Sebastian says thanks.
Thank you.
Are there any other questions from the audience? Does anybody want to raise question? If this is not the case, I think we can thank Chen again for his great presentation of XMS Viewer. I hope you like this format of presentation and that you can get something out of it. And from now on, all untargeted metabolomics projects will be processed through the XMS Viewer pipeline. And Chen has